Hello, my name is Camille Bouchard and I'm a PhD student at Dr. Tremblay's laboratory, where I also did my master's degree. Our laboratory is specialized in molecular biology and studies genetic therapies for hereditary diseases such as Duchenne muscular dystrophy, dysferlinopathy, and Friedreich ataxia. In this webinar, I will talk about my master's degree project, which was about finding the optimal mouse model to study the effect of therapies for Friedreich ataxia. First of all, Friedreich ataxia is the most common form of ataxia in the world. The cause of this hereditary disease is the expansion of the GAA trinucleotide in the first intron of the frataxin gene. Patients can have up to 1,700 repetitions, leading to a decrease in frataxin synthesis. We wanted to find the optimal mouse model to study eventual treatment for Friedreich ataxia. So we looked for one which had a comparable phenotype to human patients, but also a similar genotype. We chose to amplify the mild phenotype of the YG8SR mice using a shRNA to decrease the frataxin expression even more and evaluate the phenotype. Along this experiment, we also evaluated the phenotype of YG800 mice, a new available model developed from the YG8SR mice. Our results were a slight increase of the phenotype with the shRNA and a more comparable phenotype to the human one for the YG800 mouse model. The GAA expansion causes a decrease in frataxin synthesis, a protein which is necessary to metabolize iron. This disease is usually diagnosed around puberty, and its impact is mostly on movement coordination, but also can cause cardiac hypertrophy, which can be lethal. In our search for the optimal mouse model to study the impact of a genetic treatment, we saw previous mouse models which had either a very mild phenotype or an extremely severe one. The first models had a complete deletion of the frataxin gene, making it very severe, leading to an early death of mice. Then came mice with a GAA expansion, either in the mouse frataxin gene or in the human transgene that they were given. The most interesting mouse models for our purpose were knockout in mouse frataxin and carry the human gene. YG8SR contains around 300 GAA and has a mild phenotype, while the new YG800 is around 800 repetitions, but the phenotype was still to be analyzed. The first part of our project was to inject different shRNAs targeting the frataxin gene to decrease its expression even more in YG8SR mice, and hope to increase their slight phenotype. The goal was to have an impaired movement coordination comparable to the human one to help measure the impact of a treatment on the phenotype. This figure summarizes the different steps of the project on the YG8SR mice. At 2 months, the mice were injected with a saline solution or shRNA in the tail vein by the animal technician. 5 weeks later, 4 different coordination tests were performed to measure the movement coordination of the mice. After necropsy, the proteins were extracted from tissues such as the tibialis anterior muscle, the heart, the liver, the cerebrum, cerebellum, and dorsal root ganglions. Frataxin protein concentration was assessed by an ELISA test. Since there was a high intersample variability, we extracted RNA from the tissues and ran an RT-qPCR to quantify the expression of frataxin mRNA. We also measured the viral particles distribution by PCR on the DNA of the cell tissues. The second part was to do a comparable evaluation of the new YG800 mouse model. Since no injection was needed and we wanted to characterize an unknown phenotype, we did the same tests on the YG800 mice, every 3 weeks in mice aged from 8 to 26 weeks. After necropsy, the proteins were also extracted from the same tissues to be quantified in different ELISA tests. We quantified not only frataxin but also aconitase and troponin T, which are known to be biomarkers for Friedreich ataxia. We also quantified the length of the GAA expansion by PCR. Our hypothesis was that the longer GAA expansion increased the phenotype as it does in humans. In fact, literature shows that the longest GAA give a more severe phenotype in patients. We designed different shRNAs to target frataxin and noticed that the most effective ones both targeted the exon 4. We evaluated the movement coordination of both mouse models with 4 tests: crossing a notched beam, an inverted T-beam, walking on a parallel rod floor, and hanging to an upside-down grid. The notched beam is easier for mice than the inverted T-beam test. Mice with impaired movement coordination showed difficulty crossing the inverted T-beam with all 4 limbs on the top beam. In fact, impaired mice make many footfalls by walking on the lower part of the beam instead of the top one, with a specific gait which Friedreich ataxia mice develop slowly with time. The hanging grid test is also used to see coordination impairment. In fact, when mice have a reduced movement coordination, they tend to rapidly grip their own leg instead of the metallic grid. Our hypothesis was that the shRNA targeting the frataxin gene used on the YG8-SR mice and the longer GAA expansion of the YG8-800 mice could increase their phenotype. First, I will present the results obtained with the YG8-SR mice injected with an shRNA targeting the frataxin gene. We compared different shRNAs and show the best ones here. The SH1 and SH3 were both targeting exon 4 and gave the greatest phenotype increase as well as the greatest frataxin concentration decrease. Here we show the performance of mice injected with SH3 RNA on both beams and on the hanging grid. These tests allow to measure the movement coordination by counting the footfalls as well as the time needed to cross the beams. We also observe if the mice could grip the grid with the hanging test. And impaired coordination equals to more footfalls, more time needed to cross the beam, and less time hanging on the grid. In fact, when the mice lack coordination, they grip their own feet instead of the grid, leading them to fall down. We compared results to 2 types of control mice, which were all tested at the same time in the same conditions. We had mice injected with a saline solution to evaluate the impact of the injection stress. We also had mice injected with the scramble shRNA, targeting another gene but not frataxin. Finally, we compared 2 shRNA concentrations. Figures A and B show that injected mice need more time and make more footfalls to cross the notched beam. Figures C and D show the same on the inverted T-beam. Finally, figure E shows that they can hang for less time than the control mice. We also evaluated the frataxin protein concentration in different tissues. A significant decrease was seen in the liver, yet was only an insignificant trend in the cerebellum. We also looked for the frataxin mRNA expression, and this time the decrease was significant in both tissues, being the liver and cerebellum, since the gap between all values was smaller than for the protein. We also noticed a curious increase in the frataxin mRNA in the liver of mice injected with the scramble shRNA, which did not target frataxin, but our hypothesis is that it could target another gene which downregulates expression in the liver. Now I will present the other mouse models' results. Here we compare the YG8-800 mice with the Y47 healthy mouse model. The gap between both groups increases with time, and at 20 weeks of age, the difference is significant for all tests. Indeed, the YG8-800 need more time and make more footfalls on both beams, and stay for less time on the hanging grid than control mice. Here is more information about the YG8-800 mouse model. Figure 6 shows a significant decrease of frataxin in many tissues compared to the Y47 healthy mice. In fact, almost no frataxin is measured in muscles, liver, and dorsal root ganglions. A very low concentration is measured in the heart, cerebellum, and cerebrum, also making it significantly inferior compared to the Y47 mice. Just below, Table 1 compares the frataxin concentration in tissues from both Friedreich ataxia mouse models used in our project. We made a ratio of the concentration of the Friedreich ataxia mouse versus the Y47 mouse for both YG8-SR and YG8-800. The YG8-SR results are for mice without shRNA injection. The shRNA did not show a significant decrease of the frataxin concentration in tissues except for liver. Table 1 shows that YG8-800 have an important decrease of the frataxin concentration in all tissues, which could explain the phenotype difference. Also, we noticed a cardiac hypertrophy in YG8-800 mice, which was not present in YG8-SR mice, even injected with shRNA. It is significant both in males and females and helps us conclude that the YG8-800 mouse model is comparable to the human one for Friedreich ataxia. In conclusion, frataxin targeting shRNAs decrease frataxin expression in the cerebellum and liver and decreases slightly the movement coordination. More importantly, the YG8-800 mouse model phenotype has been characterized. It is comparable to the ataxic human one for its impaired movement coordination, frataxin concentration decrease, and especially for the cardiac hypertrophy. Therefore, our laboratory chose to use this mouse model to evaluate genetic treatment of Friedreich ataxia. This new model has a human frataxin transgene and allows to make the same genetic treatment, which could be used in humans without modifying the target sequence. Also, since the phenotype is comparable to the human one, it will be possible to evaluate the impact of the treatment on the phenotype, not only on the molecular level, which makes it even more interesting for our purpose. To summarize this research in one sentence, we compared different mouse models to find the optimal one for the purpose of evaluating a genetic treatment for Friedreich ataxia, and the YG8-800 is the most convenient one for our needs. In fact, its phenotype is comparable to the human patient ones, and its human transgene is optimal to test genetic treatments. I want to thank you for listening to my presentation and also thank Dr. Tremblay for his support, as well as our lab team for their help. I also want to thank La Fondation de Grand Défi Pierre Lavoie and Ataxia Canada for their financial support to this project. If you wish to know more about our incoming projects, here is the QR code to my LinkedIn page. Thank you.